Hey everyone, and welcome back to the HIV RNA Test Guide podcast. We're your go-to source for staying up to date on all things HIV testing and treatment. And today we're diving deep into some really exciting news
Oh, absolutely.
in the world of HIV research.
Yeah.
A potential breakthrough treatment called AGT-103T.
It's definitely a development worth exploring, for sure. For years, antiretroviral drugs have been the standard for managing HIV, and they do a great job at suppressing the virus. But AGT-103T is something different. It's a type of gene therapy that aims to modify the body's own immune system to fight HIV.
Okay, so before we get ahead of ourselves,
Right.
let's break this down for our listeners. What exactly is gene therapy, and how does it work in the context of HIV?
Good question. Gene therapy essentially involves modifying a person's genes to treat or prevent disease. In the case of AGT-103T, the focus is on P cells, which are key players in the immune system's defense against viruses.
So it's like giving the immune system a boost, specifically targeting HIV.
Precisely. The process involves extracting T cells from the patient's blood. Then in the lab, these T cells are modified using a special vector that delivers genetic instructions. These instructions essentially reprogram the T cells to resist HIV infection. Finally, the modified cells are infused back into the patient where they can hopefully multiply and help control the virus.
Wow, that sounds incredibly complex. It's like something out of a science fiction movie.
It is pretty amazing technology. And what's really exciting is that AGT-103T targets a very specific vulnerability that HIV creates in the immune system.
Oh, I'm all ears. Tell me more.
Well, you see, HIV weakens the immune system by attacking and destroying a type of T cell called CD4 T cells. These CD4 T cells are crucial for coordinating the immune response against infections, including HIV.
So by targeting these CD4 T cells, AGT-103T is essentially trying to redual the immune system's defenses against HIV from the ground up.
That's a great way to put it. AGT-103T aims to restore these vital CD4 T cells, making the immune system much more effective at fighting off the virus.
Okay, so that's the science behind it. But what about the real-world application? I understand AGT-103T has recently completed its phase 1 clinical trial. Can you tell us more about this trial and what the results tell us so far?
Absolutely. The phase 1 trial was conducted on a small group of 7 people living with HIV who were already on stable antiretroviral therapy. This initial phase of research was primarily focused on safety and feasibility.
So basically making sure that the treatment wasn't going to cause any serious harm and that it could actually work in the human body.
Exactly. And the results were very encouraging on both fronts. Firstly, the trial showed no serious side effects. The few side effects observed were generally mild and temporary things like headaches or fatigue, and mostly related to the procedures before the gene therapy itself.
That's a huge relief. But did they see any signs that the treatment was actually working, that it was doing what it was supposed to do?
Yes, they did. And this is where things get really exciting. Researchers measured the levels of those HIV-specific CD4 T cells in the patient's blood, and in all 7 participants, these crucial cells were detected after the treatment.
So the modified T cells were doing their job. That's incredible. Did they see any other positive changes in the participants?
The most impressive finding was the significant increase in the number of those HIV-specific CD4 T cells. In some cases, the increase was dramatic. We're talking a 9 to 300-fold increase compared to before the treatment.
Wow, 300-fold increase. That's a huge jump. Does this mean we can start talking about a cure for HIV?
While it's still early to declare a cure, the results are incredibly promising. The phase 1 trial wasn't designed to test whether patients could stop taking their antiretroviral medications, but the significant boost in T cell counts suggests that AGT-103T has the potential to give the immune system a much stronger grip on controlling the virus.
So it may not be a cure just yet, but it could be a major step towards a functional cure where people could potentially control the virus without needing daily medication.
That's the hope.
This is all incredibly exciting news, but I know this is just the beginning. There's still a long road ahead before AGT-103T could become widely available. What are the next steps in the research process?
You're right, the journey continues. The next steps are phase 2 and 3 trials, which will involve larger and more diverse groups of participants. These trials will focus on key questions, like whether AGT-103T can allow patients to safely stop taking their antiretroviral therapy while maintaining viral suppression. They'll also be looking at how long these modified T cells remain effective.
So figuring out if AGT-103T can provide long-term viral control without the need for daily medication and how durable the treatment effects are. That makes sense. But I imagine there's some hurdles to overcome along the way. What are some of the challenges facing researchers as they move forward with these trials?
One major challenge is the complexity of manufacturing personalized cell therapy. It's a highly intricate and expensive process.
I can imagine. We were talking earlier about how personalized this treatment is, with cells being extracted and modified for each individual patient. That's a far cry from mass-producing pills in a factory.
You got it. Scaling up production to make AGT-103T widely available is a big task. And then there are the regulatory hurdles involved in getting new treatments approved for use.
So it's not just about the science, it's also about navigating the logistical and regulatory side of things.
Exactly. But despite the challenges, the success of this phase 1 trial has generated a lot of optimism.
It definitely feels like there's a real sense of momentum building in the field of HIV research.
I agree, there's a lot to be hopeful about. Speaking of hope, I want to touch on something that's especially relevant to our listeners here at the HIV RNA Test Guide podcast, the importance of early testing.
Yes, early detection is so crucial, and we've talked a lot on this podcast about the HIV RNA test, which is a powerful tool for early detection. Can you remind our listeners about how this test works and why it's so important?
Absolutely. The HIV RNA test directly detects the virus's genetic material, its RNA, in the blood. This allows for much earlier detection of HIV compared to traditional antibody tests. We're talking as early as 10 to 33 days after exposure, significantly faster than antibody tests.
Yeah, it's a game changer, especially for those who might be worried about a recent potential exposure.
Exactly. Early detection has a number of benefits. First, starting treatment sooner can help preserve the immune system and prevent the virus from progressing. Second, it can provide more accurate results for those who may have had a recent potential exposure to HIV. And third, for those already living with HIV, the HIV RNA test, also known as a viral load test, is essential for monitoring treatment effectiveness.
So the HIV RNA test is a valuable tool throughout the entire HIV journey, from diagnosis to treatment monitoring.
Precisely. And it also plays a crucial role in clinical trials, like those for AGT-103T. Researchers use the HIV RNA test to track the therapy's effectiveness by accurately measuring how much virus is present in the body.
So it's a key part of evaluating the success of new treatments. This is all really fantastic information for our listeners. Now, I want to circle back to the challenges we were discussing earlier, specifically the cost of manufacturing these personalized cell therapies. Can you elaborate on why this type of therapy is so complex and expensive?
Of course. As we've discussed, personalized cell therapy like AGT-103T involves extracting cells from each individual patient, modifying those cells in a specialized lab, and then infusing them back into the same patient. This individualized approach is very different from producing a traditional drug that can be mass-produced in a factory.
So it's a highly customized process, almost like creating a bespoke suit.
That's a great analogy. And because of this individualized approach, the manufacturing process is incredibly intricate. It requires highly skilled technicians, specialized equipment, and rigorous quality control measures to ensure the safety and effectiveness of the modified cells.
I can see how that would make it challenging to scale up production and make this type of therapy more widely accessible.
You're exactly right. Currently, personalized cell therapies are typically produced in small batches in specialized facilities. To make these treatments accessible to everyone who needs them, we need to find ways to streamline and automate the manufacturing process while maintaining the highest standards of quality and safety.
So it's not just about the science behind the therapy itself. It's also about the logistics of making it available to millions of people around the world.
Precisely. And thankfully, there's a lot of research and development happening in the field of cell therapy manufacturing. Scientists and companies are working hard to develop new technologies and platforms to produce these therapies more efficiently and cost-effectively.
That's encouraging news. And what about the long-term safety of gene therapy? Are there any potential risks or side effects that researchers and patients need to be aware of?
That's an important question. While the phase 1 trial for AGT-103T didn't show any serious side effects, long-term monitoring is absolutely crucial. We need to follow patients over time to make sure there are no delayed or unexpected effects that might emerge.
So long-term safety data will be a key piece of the puzzle as research continues.
Absolutely. And it's also important to remember that gene therapy is still a relatively new field. As with any new medical treatment, there's always a learning curve. We need to proceed with caution and carefully evaluate both the benefits and risks.
It seems like there are several factors that need to be considered before gene therapy becomes a standard approach to treating HIV.
You're right. We need to address the cost, long-term safety, and figure out how gene therapy best fits into the existing landscape of HIV treatment. Keep in mind, we already have very effective antiretroviral drugs that can suppress the virus to undetectable levels. So it's not necessarily about replacing those treatments, but finding the best ways to integrate gene therapy and personalized treatment plans for each individual.
That makes a lot of sense. It's about adding another powerful tool to the toolbox and figuring out the best ways to use it in combination with the tools we already have. It's an exciting time to be following HIV research, that's for sure.
I agree. There's so much potential on the horizon, and I'm optimistic about what the future holds for people living with HIV.
Before we move on to our final thoughts, I want to touch on something you mentioned earlier about the HIV RNA test being used to monitor treatment effectiveness. Could you go into a bit more detail about how that works for our listeners? What kind of information does this test provide, and how does it help healthcare providers manage HIV treatment?
You bet. The HIV RNA test, or viral load test, measures the amount of HIV genetic material, its RNA, that's present in a person's blood. The primary goal of HIV treatment is to suppress the virus to a level that's undetectable by this test.
So when we say undetectable, does that mean there's absolutely no HIV in the blood?
Not quite. Undetectable simply means that the level of virus is so low that the test can't detect it. This doesn't mean that the virus has been completely eradicated from the body, but it does mean that the treatment is working effectively and that the person's immune system is healthy.
That's an important distinction. So how often should people living with HIV get their viral load checked?
It varies depending on the individual and their treatment plan, but typically people on antiretroviral therapy will get their viral load checked every 3 to 6 months.
And what happens if the viral load is detectable?
If the viral load is detectable, it means that the virus is replicating and the treatment might need to be adjusted. The healthcare provider will work with the patient to determine the best course of action. This might involve changing medications or addressing any factors that might be interfering with treatment adherence, such as difficulties taking medications as prescribed.
So it's a dynamic process of monitoring and adjusting treatment as needed to ensure the virus stays suppressed.
Exactly. The HIV RNA test is a critical tool in helping healthcare providers make sure that their patients are receiving the most effective treatment possible.
It really highlights how crucial the HIV RNA test is for both individuals managing HIV and the researchers working to develop new treatments. But let's shift gears back to AGT-103T and the bigger picture of gene therapy. What are your thoughts on the long-term potential of this approach? Do you think we'll see a day when gene therapy becomes a standard part of HIV treatment?
That's a great question, and it's tough to predict the future with certainty. But based on what we've seen with AGT-103T and other promising gene therapies, I'd say the potential is enormous. If these therapies continue to show positive results in larger trials and demonstrate long-term safety and effectiveness, they have the potential to revolutionize the way we treat HIV.
What are some of the key factors that will determine whether gene therapy becomes widely adopted?
One major factor is cost, as we discussed earlier. Right now, personalized cell therapies are very expensive to produce, which could limit their availability, especially in resource-limited settings.
So finding ways to make these therapies more affordable will be crucial for ensuring equitable access for everyone who could benefit.
Exactly. Another key factor is long-term safety. As promising as the initial results are, we need to be cautious and monitor for any potential long-term side effects.
So gathering more data on the long-term safety and efficacy of these therapies is essential. What about how gene therapy fits into the current treatment landscape? As you mentioned earlier, we already have effective antiretroviral drugs that can keep the virus suppressed.
That's an important point. It's not necessarily about replacing antiretrovirals, but rather about figuring out the best ways to integrate gene therapy into existing treatment strategies. We need to identify which patients are most likely to benefit from gene therapy and how to best combine it with other treatments for optimal outcomes.
So it's about tailoring treatment plans to each individual's needs and finding the right combination of tools for each person. Personalized medicine at its finest. It sounds like there's a lot of work to be done, but the possibilities are incredibly exciting.
I couldn't agree more. We're living in a remarkable time for HIV research, and gene therapy is just one of many exciting avenues being explored. I'm incredibly optimistic about the future and what these advancements could mean for people living with HIV around the world.
It's certainly inspiring to think about a future where HIV is no longer a life-defining illness, but a manageable condition. Before we wrap up, I want to thank you for joining us today and sharing your expertise on this fascinating topic. You've given our listeners a lot to think about and a renewed sense of hope for the future of HIV treatment.
It's been my pleasure. I'm always happy to talk about this important work.
And to our listeners, thank you for tuning in to the HIV RNA Test Guide podcast. We hope you found this deep dive informative and empowering. If you're interested in learning more about AGT-103T or the HIV RNA test, we'll have links to some helpful resources in the show notes. And remember, knowledge is power. The more we know about HIV testing and treatment options, the better equipped we are to make informed decisions about our health. Until next time, stay informed, stay curious, stay